Hello, in this lecture I will talk about RNA interference. We'll learn the mechanism of RNA interference, the process of RNA interference, and about its various applications. Before that, we'll go through a brief history of RNA. RNA was first discovered in plants in 1990. It was called post-transcriptional gene silencing. It was observed that in a purple color plant producing purple color flower, when double-stranded RNA was introduced, instead of enhancing the blue color, it resulted in white color of flower. So it was that time it was not easy to explain that one. No, there was no explanation how it happened actually, how double-stranded RNA was inducing uh, white color formation instead of increasing the color. So the first evidence that double-stranded RNA could lead to gene silencing was found in C. elegans in 1998. It was observed that when double-stranded RNA was introduced into C. elegans, it resulted in gene silencing. In the same year, 1998, in Drosophila also, when double-stranded RNA was injected into the embryo, it resulted in the gene silencing. And later on, similarly in mammalian cells, it was found that use of 21 to 23 nucleotide siRNA activate RNA, but not antiviral response in mammalian cells. So in 2001, RNA interference was observed in mammalian cells. And in 2003, using RNA interference, systematic functional analysis of the whole C. elegans genome was deciphered. In 2004, therapy for polyglutamine-induced neurodegeneration caused by ataxin-1 gene in a mouse model of spinocerebellar ataxia was observed. So it was actually the first in 2004 therapeutic potential of RNA was discovered. In in the same year 2004, November 2004, RNA was used for treatment of lowering of cholesterol. In 2004 and 5, phase 1 clinical trials initiated for age-related macular degeneration. In 2008, Equity Pharmaceutical finished phase 2 studies and started phase 3 for age-related macular degeneration. And in 2018, first ever FDA approved RNA-based drug was approved in 2018. So this is a brief history of RNA. So the RNA was discovered by Fire and Mello in 1998, and this was published in Nature, journal Nature. So when double-stranded RNA was injected into C. elegans, it resulted in gene silencing. As you can see in this picture, MAX3 mRNA. So this is negative control where there is no expression of MAX3 mRNA. And here you can see this is uninjected, means this is expression of MAX3 mRNA. And when antisense RNA was introduced, there was some gene silencing of MAX3 mRNA expression, and when antisense RNA was used. But when double-stranded RNA was introduced in the C. elegans, there was a complete inhibition of MAX3 mRNA. So this was the first incident that double-stranded RNA can induce gene silencing in C. elegans. So this is basically the discovery of RNA. However, in mammalian cells, when double-stranded RNA was introduced into mammalian cell, it was resulted in the global non-specific gene silencing. Instead of specific gene silencing, it resulted in global non-specific gene silencing, which is called interferon response. So, and it was not suitable there. So, but in 2001, Al Bashir et al. Published one paper in Nature. They use 21 to 23 short double-stranded instead of a full-length double-stranded RNA. They use a short double-stranded RNA, that is 21 to 23 double-stranded RNA. So when they used this small double-stranded RNA, they could see almost 90 to 95% inhibition of the gene expression. As you can see in picture here, This is this control here. This is a non-specific siRNA, and this is the specific siRNA, that is laminin specific siRNA, which resulted in complete gene silencing, as you can see with the reduction in the color. So in the how actually this RNA works in mammalian cells, when 21 to 23 base pair siRNA is introduced into cells, it get associated with a RISC complex, gene silencing complex, which is called RNA silencing complex. And then the sense strand is separated, and the antisense strand then hybridized with the RNA, and then this RNA gets degraded, and then gene is completely silenced then. So here we can see the example here. This is GAPDH, expression of GAPDH in mammalian cells. When the GAPD specific siRNA was introduced, it resulted in a complete knockdown of the GAPDH gene expression here. As you can see, there is no green color. Green color here is the GAPDH expression. It's protein, or GAPD is protein here. And when this siRNA, which is specific for GAPDH, was introduced into mammalian cells, it resulted in inhibition of GAPDH expression in HeLa cells. So this is basically the mechanism here. Synthetic gene specific siRNA, we have to generate specific gene specific siRNA. Then it is introduced into the cell, it crosses the cell membrane, and then siRNA is delivered into the cells. siRNA then enter into the RISC, that is RNA silencing complex, and then it's unzipped. So passenger strand, that is sense strand, is separated, and the antisense then make complementary pairing with the, it hybridizes with the target mRNA, and then it degrade the specific mRNA, and this result in the gene silencing. So target mRNA cleavage and degradation occurs, and then the gene is silenced. So this is the mechanism of RNA. So what is the significant properties of RNA, why it is so important for us? Double-stranded RNA, not single-stranded RNA, is the interfering agent. So double-stranded RNA is more stable as compared to single-stranded RNA. So it is more potent as compared to single-stranded RNA. Sequence-specific loss of mRNA protein, the loss gene expression silencing is a highly sequence-specific. It doesn't causes loss of expression of other genes. It is highly potent. Few double-stranded RNA molecules per cells are effective, or at least 10 to 100-fold more potent than antisense RNA or ribozyme technology. So it is very potent. It is very specific also. A single base pair difference is enough to block the process. If there is a single base pair difference, the RNA interference won't occur. So it will block the whole process. So it needs the exact sequence homology. Its precise mechanism of action reduces the potential side effects. So its mechanism of action is very precise. It reduces the potential side effects. So this is much better than other chemical drugs. RNA can be exploited to silence several genes simultaneously. So this is another, like in case of cancer, where several genes get mutated. So RNA can be exploited to silence several genes simultaneously. Inhibiting multiple targets simultaneously reduces the probability of developing drug resistance. Suppose if you want to use RNA as a drug, then inhibiting multiple targets simultaneously reduces the probability of developing drug resistance, as we see in other drug, antibiotics and others. So how to produce this siRNA? There's several methods like chemical synthesis, in vitro transcription, or endogenous expression using plasmid or viral vectors. So chemical synthesis is very fast, but it requires it's more expensive, and it is used for transient gene silencing. So it's not a stable gene silencing. Only for a short period of time gene can be silenced, but it is expensive. Other method, in vitro transcription, you can generate several pool of siRNA and higher probability of reducing off target, and also so this also is low cost as compared to the chemical synthesis. The most preferred method is plasmid or use of plasmid or viral vectors to generate saRNA or shRNA, short hairpin RNA. So these allow both long-term and transient gene silencing. So these vectors, plasmid or viral vectors, which has siRNA codes, They can be used for both long-term and transient gene silencing, and also inducible gene silencing can be used. So these are the more preferred method of producing siRNA, as it can be used for long-term gene silencing. So what are the various application of RNA? RNA has several applications now. It can be used as a genetic tool. It can be used for faster identification of gene function. If it is a new gene has been discovered, so to know what is its function, RNA can be used. Functional validation of novel gene. Novel gene can be validated. Their function can be validated. Loss of function phenotype analysis can be done. Drug development, drug target validation can be done using RNA. And RNA itself can be used as a drug. So any gene is a potential target for silencing by RNA, and hence the therapeutic possibilities are endless. So any gene can be used as a potential target for developing drugs. It can combat viral infection like HIV, RSV, hepatitis. Cancer and neural disorders can be treated using RNA drugs. So there are several applications of RNA. So role of RNA in a drug discovery, how RNA can be exploited in drug discovery. Faster identification of gene function as compared to knockout mice studies. Functional validation of novel gene in cancer and other neurological disease and other diseases. Help in new drug discovery from target validation to preclinical animal studies, proof of concept studies. RNA can be exploited to silence several genes simultaneously that will help in understanding more clearly those conditions that result from multiple gene defects such as type 2 diabetes, autoimmune disorders and cancer. So this can be is a very good tool actually to basically understand very complex disease like diabetes and cancer, because several genes can be silenced simultaneously and they can be studied. Their effect can be analyzed. Implication in disease that are prone to developing drug resistance by simultaneously disabling multiple targets. So drug resistance can be overcome by using RNA based drugs. Loss of function phenotype analysis, tissue specific knockdown for individual genes, its impact on transcriptome, proteome, metabolome, and overall genotype and phenotypic alternation. So basically tissue specific knockdown also can be done using RNA based drugs. Drug target validation, how it is better as compared to traditional approaches. Traditional approach for drug target validation is dependent on costly, labor intensive and time consuming screens. RNA based inhibitors can be generated much more readily and can facilitate much higher throughput to address the challenge of drug target validation. So this is easier as compared to traditional approaches. RNA can validate any drug target in a very short time. And so now the most important application of RNA is RNA as a therapeutic agent. Okay, so any gene is a potential target for silencing by RNA, and hence the therapeutic possibilities are endless. So it means that any gene, as any gene is can be silenced by RNA. So those genes, so there it means there are several targets can be used in case if we use RNA as a drug. So at present, we have three different platforms like small molecules. So this is a major product platform in pharmaceutical industries, and more than 1,000 approved products are there in the market. So small molecules at present, the market is dominated by small molecules which are chemically synthesized or obtained from plant. So more than 1,000 is approved. Another platform is protein and antibodies. These are also used as a drug. So major product platform in biotechnology industry. At present, more than 250 are approved, which are in the market. So RNA is a new one. This is a potential new product platform. RNA, natural process for gene silencing, potent and selective, as there is a precise mechanism of action. Because the precise mechanism of action is highly specific, so side effects are less as compared to small molecules. Widely used research tools, already being used as research tools. At present, situation is that around around six drugs, siRNA-based drugs, have been approved by the FDA. So we'll see in the end of this lecture what are those drugs which have been approved by FDA. So this is just to show various difference between the siRNA based drugs and the small molecules. siRNA based, they are highly specific because it's a sequence driven. Small molecules specificity is very low. Potency typically in picomoles, they are highly potent and here the potency is very variable here. Number of axis targets are more than 1,000. Actually, sRNA can target several different genes. So there's several targets, more than 1,000 targets are available. Here, for small molecule targets are very limited, 500 to 1,000. Number of potential leads and backups. So you can generate several leads in the very short span of time. Like more than 10 to 100 leads can be developed, potential leads can be developed. But in case of small molecule, it's less than 2 or 3. From 2 or 3 means you don't get many molecules to take for clinical trials and all that one. Speed to lead molecules, it takes around 4 to 8 weeks. So in a very short span of time, you can get a lead molecules for clinical studies. And in case of small molecules, generally takes 2 to 4 years to go to from preclinical to clinical stage. So it takes quite a lot of times for developing a small molecule based drugs. Species cross reactivity, it is high as compared in this one. In case of a small molecule is low. Manufacturing, it is very common, rapid and scalable method. So it's very easy to manufacture siRNA and large scale also at commercial level, and it's variable and can be very complex. Small molecule synthesis of small molecule is very complex, and moreover, siRNA based drugs they have less toxic as compared to small molecules. So this is RNA in adult mouse. How so this just to show the therapeutic potential of RNA. So here is the reporter luciferase gene reporter. Here you can see the color here, green color here. This is the expression of luciferase in the mouse tissue. When the luciferase specific siRNA was introduced, it resulted in a complete inhibition of the luciferase expression here. This is just to show that RNA can work very well in the adult mouse also. And here is the control. So RNA has a tremendous potential as a therapeutic for neurodegenerative diseases, as we know that there are very few drugs available for treatment of neurodegenerative diseases. So as siRNA based drugs can play a very important role here. For example, spinocerebellar ataxia type 1 (SCA1) is characterized by ataxia, cerebral atrophy, and loss of cerebral pumpkin cells, and formation of intranuclear aggregate containing the disease protein. At present, there is no effective therapy. Only inhibition of mutant allele is the best approach, which can be earlier was tried with antisense technology. Antisense and ribosome-based technique failed in animal models, so they worked in tissue culture model, but animal model they failed. So RNA was used. Now the RNA-based drug was used to inhibit the neurodegenerative caused by mutant ataxin-1 in a mouse model of SC1, and it was found that upon intracellular injection, siRNA directed to ataxin-1 profoundly improved motor coordination, restored cerebral morphology, and resolved ataxin-1 inclusion in Parkinson's cells SC1 mouse model. So this work was published in Nature Medicine by Zia et al. In 2004. So this work clearly showed that RNA can, as compared to antisense or small molecules, RNA-based drugs can be used for treatment of neurodegenerative diseases. So this was just one example to show that it is a very good strategy. So this is another example, RNA treatment to lower cholesterol. So this was published in Nature 2004. Apolipoprotein, ApoB is essential for formation of low density lipoprotein in metabolism of dietary and endogenous cholesterol. ApoB protein is non-druggable target as it is not amenable to inhibition by small molecules or protein-based therapeutic. So ApoB protein cannot be targeted by small molecules or any protein-based therapeutics. So basically, there is no drug for which can target ApoB protein. So here, siRNA can play a very important role. siRNA targeting ApoB was chemically stabilized using cholesterol to prevent degradation by exo and endonuclease in the serum tissue. So this was very important strategy they employed here for controlling for lowering cholesterol. So they conjugated they chemically stabilized the ApoB siRNA, so then which prevented it made it more stable. So it was not degraded by exoendonucleases in the serum and tissue. So so it means siRNA more of the it was as efficiently targeted to the tissues. So intravenous injection of SRN result in silencing ApoB mRNA in the liver and jejunum and decrease plasma level of ApoB protein and reduce total cholesterol. So this work also showed that RNA is very effective in animal models for controlling for reducing cholesterol. But however, there are few challenges for RNA-based therapeutics. The main obstacle is delivery of functional siRNA to the target tissue. How to deliver siRNA so that there is less degradation and it reaches the target tissues. Minimize digestion by nucleases. Extend blood half-life. Cross cell membrane. Achieve adequate target tissue specific bioavailability. So bioavailability should be more so that it can be more potent. Assess siRNA delivery non-invasively without injection if it is can be given oral or by inhalation. Prevent activation of immune response and off target effects. So these are some of the challenges which needs to be overcome to develop SRN based drugs. So this is one major issue when we use siRNA as drugs. So it may lead to activation of mammalian immune system. So it has been that siRNA can induce cytokine expression. So siRNA without carriers do not activate or at least poorly activate pro-inflammatory cytokine in vitro and vivo. In complex with cationic carriers such as liposome specific sequence such as this one, within the siRNA duplex are immunostimulated both in vitro and vivo. siRNA activates cytokine primarily by binding to TLR7 and 8 in acidic level. So these are the basically these siRNA can binds to TLR toll-like receptor 7 and 8 in endosomes. TLR here you can see here TL 7 and 8. So it binds to this one and that activates cytokines here. So this is the main problem, main challenge actually which needs to be overcome. So basically the activation of cytokines by siRNA. So this is the slide just to show that how RNA can be therapeutic potential of RNA can be harnessed. So chemically, either we can do chemically synthesize siRNA which can be modified, chemically modified. Naked siRNA can be used, or any shRNA can be used. So all these can be used. They can be then they introduce in the cell. Then it binds to the RNA RISC complex, RNA-induced silencing complex, which is basically comprised of AGO2 protein, argonaute protein. And then the sense strand is removed and antisense RNA binds to the target mRNA, and then target mRNA cleavage occurs, and which leads to target-specific gene silencing. But the another problem which may occur actually is the if siRNA enters the endosomes here, it can activate TLR7 and tau-like receptor 7 and 8, and then leads to expression of cytokines like NF kappa B and interferons. Okay, so which can lead to side effects. So this needs to be pathway should be overcome when we are using siRNA as a therapeutics. So another important concern is that how to deliver the how to carry out the systemic delivery of siRNA. So there are some barriers for when we carry out systemic delivery as siRNA. There are 5 major accessible barriers and nanoparticle delivering siRNA must overcome to reach its target cells. So these obstacles are interaction with blood components. It can interact with blood component, protein and all that one. And trap them with capillaries of lungs and tissues, so it won't reach the target tissues then. Uptake by RAS and by other phagocytic cells. If it is taken up by phagocytic cell, it is also get degraded. Filtration by kidneys, so kidney can just filter it out and it won't be then bio available, and then extravasation from blood to the target tissue. So these are the some barriers actually for systemic delivery which needs to be overcome by using suitable carriers or delivery agents. So some new strategies have been devised for cell type specific siRNA delivery in vivo. A variety of strategies have been developed for the cell type specific delivery of siRNA. The delivery of siRNA is facilitated by directly conjugating the siRNA to the targeting ligands like cholesterol, aptamers. Okay, so non-covalent binding of siRNA with the highly positively charged motif conjugated to an antibody or to facilitate delivery to cells that bear the appropriate cell surface molecule. So antibodies can be used like here, which are specific to some cell surface receptor. So when we use this antibodies, this carriers siRNA, which is entered in liposome, having these antibodies specific to cell surface receptor, then it will bind to the specific cells and then it will introduce the cells to a specific tissues. Then, so these antibodies can be used exploited for cell type specific siRNA delivery in vivo. So delivery of siRNA can be delivered to patients as in a systemic way or ex vivo delivery. In systemic delivery, double-stranded siRNA is packaged into delivery vehicles like targeted nanoparticles, polymer or liposome. It is then given intravenously via inhaler or direct injection into the target tissue, the eye or tumor. So it can be. So this is how the systemic delivery can be achieved here, as it's shown here. Or ex vivo delivery, actually, this is the ex vivo delivery here. Ex vivo cell or dendritic cell hematopoietic stem cells are extracted from the patient and then transfused with the virus containing shRNA. And then these genetically modified cells are then infused into the patient. Then these again are, when they are modified here, genetically modified using viral vector containing siRNA, and then these genetically modified cells are then introduced into the body. So this is how ex vivo delivery can be achieved. So both studies can be applied to introduce siRNA systemic delivery as well as ex vivo delivery. So there at present, so these drugs have tremendous potential. So the FDA has recently approved around 6 drugs till now. The first drug was approved by FDA in 2018. So this is on petro. It was transthyretin. It was targeted as mainly TTR mRNA transthyretin, and delivery vector was lipid nanoparticles. Okay, so this was used for treatment of hereditary transthyretin mediated amyloidosis with polyneuropathy. The other drugs were second was approved in 2019. So 2019, and recently in 2024 was the sixth drug was approved by FDA. So these are the 6 different drugs which has been used, which have been approved by the FDA. And most of the drugs, Galnac was used as a siRNA was conjugated to Galnac. Galnac was used as a delivery vehicle actually, and in all these drugs. So till now 6 FDA approved RNA based drugs are available in the market. So all the FDA approved RNA drugs so far target the liver. Galnac conjugation is the dominant delivery strategy. RNA has moved from rare disease, cardiometabolic, hematology. CNS lung tumor targeted RNA is still mostly in the clinical trials. As of now, all FDA siRNA drugs are from Alnylam Pharmaceutical, which basically build the RNA based drug playbook. So now RNA has a tremendous therapeutic potential for treatment of various neurodegenerative diseases, cardiovascular diseases, viral infection, cancer, and also RNA can be exploited in stem cell therapy. So this, as you can see, so this RNA based drugs has tremendous therapeutic potential, but a lot of work needs to be done so that systemic the potency of these drugs can be enhanced without any activation of the cytokine response and reduce the side effects.